Hello, everybody. Welcome to this spatial biology masterclass. Today, we are having this masterclass with the OracleBio team. And let me present them quickly. So we have Karen McClyman. She's an image analysis project manager. And we also have Gabrielle Haynes-March, who is an R&D project manager. They will be both presenting 2 case studies on how they are using Visiopharm software to support their image analysis workflows and trust their data. The session will take around half an hour, and at the end there will be a Q&A session. So you are all invited to write down your questions in the chat, and we will take them at the end and discuss. So I hope you enjoy the session, and let's finally get started.
Thank you for the introductions. I'm going to start by giving a brief overview of OracleBio, who we are and what we do, before we move on to discuss a couple of case studies where we'll go into more detail on how we use Visiopharm to support our image analysis workflows. OracleBio is a contract research organisation specialising in digital pathology, and we undertake a range of image analysis studies to ultimately generate relevant and accurate data. We use client-specific images to develop our algorithms, which are tailored to the individual project needs, with a view to quantifying the staining and producing high-quality data readouts. We have an ever-growing team of expert image analysts supplying industry-leading image analysis services to pharma and biotech worldwide. We cover all aspects of the R&D pipeline, starting with target identification in early discovery, through to preclinical animal model outputs, exploration of translational biomarkers, and then finally covering image analysis for live clinical studies. We use a purpose-built AWS cloud infrastructure that allows users to spin up multiple CPUs or GPUs simultaneously. And the subscription model we have with Visiopharm allows us to maximize the productivity of our IA team to generate data quickly and efficiently. We're charged per image imported into the software, meaning in combination with our cloud computing-based deployment, we can have members of our IA team launching multiple instances of Visiopharm. So in theory, we can have a single user training an algorithm on one instance, running analysis on a second, and using a third instance to create training annotations. This enhances the rate at which we can complete Visiopharm-based projects, as we are not limited by individual licenses, and we can massively scale the work we do in terms of image QC and algorithm development to fit the unique needs of client projects. The case studies we'll talk about next have thoroughly benefited from this subscription model due to the massively parallel processing powers we can achieve with the IT infrastructure that's in place for high throughput analyses. And I'll now hand you over to OracleBio's R&D project manager, Gabrielle, who will talk you through our first case study on a pathologist-validated image analysis workflow.
Thanks, Karen. So I'm going to present this first case study, which showcases our involvement in this multidisciplinary triple helix INSIGHTS project and how we established a robust image analysis workflow to analyze a large number of colorectal polyp samples. So I'll start by giving a bit of background about the INSIGHTS project. INSIGHTS stands for Integrated Technologies for Improved Polyp Surveillance, which is a Glasgow University-led collaboration with the UK's National Health Service and an array of commercial partners, including OracleBio. The main goal of this project is to improve polyp surveillance in the NHS so that patients with higher risk of developing recurrent polyps further down the line can be identified, screened, and treated before these polyps become malignant. This new risk score will be developed using a multi-omics approach, including data not only from digital pathology, which is what I'm going to present today, but also genomics, transcriptomics, and also the patient's clinical history. OracleBio has been a member of the INSIGHTS consortium for the last 4 years, and we are the work package leads for the digital pathology effort. For the purpose of delivering this data, we developed a series of deep learning algorithms to generate quantitative data from over 2,200 Chi-67-stained whole slide images. And throughout the whole process, in order to ensure that the data we generate is accurate and truly represents the underlying biology of the sample, our internal pathologists help us throughout the deep learning app lifecycle, from generating training annotations all the way to assessing app performance, creating ground truth annotations, and so forth. So in terms of the digital pathology work package, the two main deliverables that we were assigned were, firstly, the generation of summary data, and that's per image, including area readouts for the main regions of interest and positive and negative Ki-67 counts split by ROI. And also the generation of cell object data, again, that's on a per image basis, including the spatial location of each cell, that is the XY coordinates, but also its assigned phenotype and a variety of morphological readouts, such as circularity, convexity, cell area, and so on. Also, due to the huge number of images in this project dataset, establishing an automated, scalable, and high-throughput image analysis pipeline was a must. So parameters like processing speeds, robustness, and by robustness, I mean the ability to handle unexpected inputs to our pipeline, and I'll give you a couple examples in a few slides. So I was saying speed, robustness, these are parameters that are key for building a scalable analysis workflow. So with this in mind, the proposed workflow follows a top-down approach and has four main components. The first one, which is a very low magnification tissue find and QC step to identify the tissue of interest and also remove any images with generalized issues. Next, still a low magnification, a deep learning classifier to separate areas of adenoma and healthy tissue. After that, we have a medium magnification around 10x, another AI app to segment the epithelial glands from the surrounding stroma. And finally, a cell detection app based on the Visiopharm bright field nuclear segmentation deep learning app, tailored to the project images to improve its performance. So on the next slides, I'm going to walk you through each step of the workflow, showing you some examples of the output generated and also the pathologist validation processes used throughout. So as I said, the first step of the workflow is the tissue detection and QC. So for tissue detection, we use a very simple thresholding app to detect the tissue of interest. And then a second app, which is deep learning based, to segment and remove the polyp stock, which has very little pathological interest. This deep learning app was built to also detect certain quality issues with the images, and I'll show you a few examples in the next slide. But here on the right-hand side, we have the polyp tissue in green and then the stock in orange, which is removed and not used for any subsequent analysis. So in terms of quality control, in a small-scale study with a few dozen images, checking every single image for large, obvious artifacts is something doable, but not here. When you are faced with thousands of samples, this initial screening needs to be robust and needs to pick up major artifacts that will impact any analysis further down the line. So for example, on the left here, we had some samples that had no hematoxylin stain, probably due to an issue with the autostainer, which means our cell detection app further down the line would have not performed as it should have. So images like this with bad stain distribution are picked up and removed in this step. On the right-hand side, I'm showing an example of slide where the scanner lamp failed, which means a number of tiles were completely missed. However, in this particular case, over 90% of the tissue is still present and in good condition, but our app flagged this sample for manual review. Also, issues with non-uniform background illumination, for example, both within the sample and across different slides were also tackled so that only the tissue of interest was segmented and passed on to the next step of the workflow. So then the next step was the adenoma classifier, which was a deep learning app trained on a number of examples covering the main polyp pathologies in the patient cohort. It was trained to recognize adenoma and non-adenoma, but also background and artifacts, such as out of focus, tissue folds, and stain deposits. Here on the right-hand side, you can see an example of the classifier overlay. So in orange, we have the adenoma. In green, the non-adenoma or healthy tissue. Then we have a bit of red, which are the artifacts. And I see there is a bit of tissue folds and some stain deposits as well towards the edge of the sample. And also glass is detected. And as I mentioned, the stock is the green hatched area here, which is removed and not used for any subsequent analysis. So here I'm showing a few examples of the pathologist-led validation we performed throughout the course of the study. So in this particular case, for the adenoma classifier, we selected 10 images at random, and on each of them, we drew two fields of view of approximately 2 millimeters squared. Then we asked one of our pathologists, who was blinded to the classifier training and performance, to annotate for adenoma and non-adenoma within the ROI boxes. Then we ran the classifier within the same areas and calculated the overlap between the pathologist ground truth and the classifier overlay for each class, both adenoma and non-adenoma. And then based on these readouts, we were able to calculate the dice score for each class and for each image. So then once we got the readouts, we averaged them all. So we achieved on average a dice score of 0.88 for the adenoma class and 0.91 for the non-adenoma class, which is well within our quality standards. The next step of the workflow is the epithelium classifier. And the purpose of this step is to separate the epithelial glands and the surrounding stroma. But before we run the app, we apply some pre-processing steps to the ROIs generated by the adenoma classifier so that we could subdivide the adenoma region into luminal adenoma and basal adenoma. Luminal adenoma, which is shown in blue here on the right-hand side, is the region of adenoma that projects into the lumen of the bowel, and there was certain interest in separating the adenoma class into these two subregions, so then separate readouts could be generated for the luminal and the basal. The basal is shown in orange, and then we have the green, which is the non-adenoma. So we generated these bands, which is 1 millimeter from the edge, and then from there, we run the epithelial classifier on all three regions separately. So we could get epithelium and stroma for each of the adenoma classes. That is the luminal adenoma, the basal adenoma, and the non-adenoma. So here in this busy picture on the right-hand side, you can see the epithelium glands in the luminal adenoma, which is the pale green, the basal adenoma in gold, and then in the non-adenoma in the darker green color. Same for the stroma in purple, light blue, and brown. And then similarly, as with the previous classifier, any areas of background or artifact that are picked up at this resolution are added to the overall background and artifact classes, which are common for all the stages of the workflow. And here I'm just showing a zoomed-in red region with the segmentation of the glands surrounded by the stroma. And as you can see, the green and purple overlays, which belong to the epithelium and stroma for the luminal band, then give way or transition into the blue and yellow as we move past this 1 millimeter mark for the luminal adenoma. There is some artifact as well in red, which looks like a small region of autofocus in this case. And finally, we reach the last stage of the workflow where we do nuclei detection and quantification of Ki-67 positive cells. This app is based on the Visiopharm bright field nuclei detection app, which is one of the deep learning apps included in the AI module. To make it more robust and to adapt it to the tissue type and stain profile used in this study, we added some manual cell annotations to the training, as the out-of-the-box app was struggling to segment cells in densely packed regions, for example, epithelial cells in the adenoma region. So we trained the app further until we were happy with the results. And then to determine the positivity of Ki-67, we simply used a threshold on the DAB channel at post-processing. So here on the right-hand side, I'm showing an example of the resulting segmentation and quantification, colored according to the ROI the cells belong to. So in the reds and blues, we have the positive and negative Ki-67 cells within the adenoma region, and then pink and green is for the non-adenoma counterpart. And same as with the adenoma classifier, we validated the performance of the cell detection and quantification app using one of our internal pathologists. So for this, we selected 3 small fields of view on 5 random images from our validation dataset. Then we asked them, one of the pathologists, to place a blue dot on every nuclei they consider to be negative and a red dot on every cell they regarded as positive for Ki-67. And then we counted them. Again, as before, the pathologists did not have eyes on the actual cell segmentation app, so they were blinded to the performance of our cell detection algorithm. Next, we run the app on the same fields of view, and we produced a readout with the number of positive and negative cells detected. And finally, we compared the numbers between the manual and the app counts. So in the first plot on the left, we can see the comparison of Ki-67 positive cells detected by the app, which is the line in purple, and by the pathologist in blue. In general, the correspondence between counts is very good, with only slight discrepancy in image 5. And we checked this image and we observed that one of the fields of view was on top of a very cell-dense epithelial gland, and the app merged together a few of the nuclei, whereas the pathologist could separate them better, and hence the difference. Then on the right-hand side, we can see the correlation between both the manual and automatic counts for the positive cells. And again, image 5 seems to be skewing the data slightly, but the correlation coefficient is still pretty high at almost 0.93. Okay, so once we were happy with the performance of our apps and the workflow setup, we were ready to batch process all 2,000 odd images and generate data. Our end-to-end workflow was composed of 8 apps, which took about an hour to run, give or take, depending on the size of the tissue. And as I said, we had 2,211 images to process. But thanks to our AWS scalable IT infrastructure and our subscription model with Visiopharm, we were able to launch 75 processing nodes, all in parallel, all with high-spec GPUs, to process all images in a truly record time. And to put it in perspective, about 2 years ago, we processed a similar dataset from the Insights project, same number of images, it was just a different biomarker. And at the time, we were using our high-performance server, which had 5 GPUs. So processing these images non-stop, 24/7, it took us about 18 days to complete the whole analysis. With our current auto-scaling capabilities, all processing was done in just under 2 days, or 45 hours, which is quite an achievement considering the size of the dataset and the complexity of the analysis. And so with this, I'll pass back over to Karen, who will walk you through the second case study.
Thanks, Gabrielle. The second case study we're covering today is from a recent collaboration with Akoya Biosciences. We received 2 mouse samples. One was healthy brain tissue and the other was a glioblastoma brain tissue. They were both stained with a multiplex immunofluorescence panel, which we used to perform spatial phenotyping. For this collaboration, we aimed to showcase the capabilities of the Visiopharm software using ultra-high-plex stained images. We approached this by developing multiple algorithms on Visiopharm to enable us to classify the tissue, to segment and then phenotype morphologically diverse cell subtypes, and also to perform area quantification of certain markers. By exporting the spatial phenotype data generated at OracleBio, we show how this can be used for downstream bioinformatic analysis to identify spatial signatures unique to the glioblastoma sample, which in turn could be investigated further in a more specific staining panel. This poster is available on both the OracleBio and Akoya websites if you're interested in reviewing this in more detail. From a top level, the poster details how the samples and images were prepared. It explains the PhenoCycler fusion workflow, which was used to perform the ultra-high-plex staining. The PhenoCode discovery panels were used, which are ideal for initial discovery work. The poster also covers the specific neuro panel used for this analysis. Akoya offer these ready-to-use panels covering the major hallmarks such as immune profiling, tissue architecture, and immune activation panels, which can be combined to investigate the more specific questions that you might have for your samples. For the purpose of this analysis, we focused on generating data for some of the main marker subsets, including the immune cells, microglia, and astrocyte phenotypes, which the next slides will show in more detail. The Akoya panels contain the most critical markers necessary to facilitate the most common analyses. And first up, we have the structural panel consisting of the markers highlighted here. This panel is important for analyses to identify areas of interest such as tumour versus healthy tissue, or to identify the vasculature. For the classifier we developed, we used markers from this set to help differentiate between the healthy and tumour tissue on the brain samples. The microglia panel consisted of the highlighted markers here, was used to identify and phenotype the microglial cell population, and this gave information regarding their activation state and their contribution to tumorigenicity. The neuron panel wasn't used for the analysis we performed, but these markers could be used to identify the mature neuron population and to perform similar phenotyping as before with the microglial cells to assess their activation state. Finally, the immune marker set was used to identify immune cell subsets such as T cells, and it was used to look for expression of markers such as PD-L1, FOXP3, and Ki67. This slide gives an overview of the standard image analysis workflow we follow at OracleBio. For this collaboration, Akoya provided an image QC report that highlighted specific markers that were presenting with a low signal-to-noise ratio and that were therefore excluded from the analysis. OracleBio also performed a visual QC of the images, where we assessed the marker staining channel by channel using the color adjustment menu within Visiopharm to easily visualize the stain quality and identify any staining artifacts that might have hindered downstream analysis and phenotyping. Any markers flagged at this point were excluded from analysis, and any major artifacts were manually removed. I'll cover the remaining steps in the following slides, where I'll go into more detail on how areas of interest were identified and how cells were accurately detected and quantified. The color adjustment menu has streamlined the QC process of high-plex images, creating more accessible and manageable interactive tools to assess stain quality and co-expression of the markers. By toggling channels on and off one by one or in combination, we can assess stain quality, stain specificity, and also identify any channel-specific artifacts, while also ensuring we're not seeing any significant bleed-through or autofluorescence in any of the channels. Using the group feature, we can also easily create channel groups for the markers we will be working with frequently, such as the microglia subgroup or the tissue architecture subgroup, which makes the annotation and review processes much easier. Here we can see an example of visualizing a single marker, GFAP, for identifying astrocytes, which we reviewed in order to assess the best analysis strategy for such markers. In the case of this project, assessing GFAP indicated it may not present the most optimal case for object detection and segmentation due to the density of the cells, which indicated the requirement for a different strategy, which is discussed later in the presentation. Typically, we develop custom deep learning algorithms for tissue classification to enable us to account for variation in stain intensity and tissue heterogeneity. However, in this case, as we only had one glioblastoma sample, a simple threshold app was developed to effectively classify the tissue into healthy and tumour regions of interest. Once we moved on to the development of the cell analysis algorithm, we noticed a distinct behaviour of cells in the healthy classified tissue directly adjacent to the tumour tissue, which was of interest and we felt would benefit from further segmentation of the tissue to allow for separate quantification of the dynamics observed in this region. This highlighted one of the main benefits of using Visiopharm, which is the flexibility we have with app design, and we were easily able to add a dilation step to the tissue classifier from the border of the tumour into the healthy tissue to capture this unique region of interest, which is defined here as the invasive margin. We identified 4 possible cell segmentation approaches we could take for analysis of the sample provided. Firstly, to identify the immune cell population. Secondly, the microglia cell population. Thirdly, astrocytes. And fourth, the neuronal cells. We used the top 3 to demonstrate the capabilities of Visiopharm when analysing high-plex samples. First up, development of an algorithm for immune cell analysis. For this, we started with the nuclei segmentation base app provided within the software, which is another great benefit of using the Visiopharm software. This algorithm can typically identify nuclei effectively in most samples, and we can easily add more training to fine-tune the algorithm to fit the samples we're working with. Alternatively, we follow the same principle as the base app, but create algorithms from scratch to include additional channels in our training, such as Ki67 or CD3, to create a more accurate detection and segmentation of such cell lines. The base app here was identifying and segmenting cells well, so we used this as a starting point to allow us to form cell objects from the DAPI channel to be used for downstream phenotyping of the relevant immune cell markers. Here's an example region of IBA1 staining in the tumour sample, and upon review of this channel for microglia detection, we noticed there was significant morphological diversity of the microglia. Had we used a traditional DAPI-based approach to capture these cells, we would have missed IBA1 positive cells where the nuclei were out of plane of the section and would not have accurately captured the whole cell object, which is important for accurate phenotyping of this cell type. In comparison, this is an example of IBA1 staining of the healthy tissue region of interest, where there is a much greater frequency of microglia with a ramified structure. Had we treated both populations of microglia the same, we would have failed to accurately capture and segment all microglia. Another benefit of Vis is that we can handle both populations individually, allowing for a more accurate segmentation profile. Shown here is the GFAP staining for astrocytes in yellow. The original plan was to perform detection and segmentation of individual cells to capture this staining, but as we can see from the density of staining, it would be challenging to accurately segment cells and capture the associated astrocyte staining. We therefore performed an area quantification of GFAP for each region of interest and assessed co-expression with Vimentin to evaluate the difference in activity states. Using the Explore QC module, we can import object data generated in the workflow and begin to assess stain quality and variation per image, per ROI, and per marker. We can use the tools available to gain a better understanding of any interesting dynamics occurring. Outliers can be identified in particular marker channels, ROI, or images analysed to give a heads up on any potential variation observed. On this plot, we're looking at the mean intensity of CD3, CD8, and Ki67 per ROI. This can help assess whether thresholds will prove effective across the whole image. We can also highlight individual objects on the interactive plots to view them on the image and assess whether they are true positive cells. Here we can look at what appears to be an outlier, but is in fact a very intensely stained true positive Ki67 cell. We imported the cell object data into Phenoplex for a streamlined process of fine-tuning thresholds per marker. An image cell panel is shown here for CD3, CD8, FOXP3 and Ki67. In combination with the colour adjustment menu, we can easily review thresholds and adjust them using the histogram slider to ensure we are detecting true positive cells. Shown here is CD3, and we can fine-tune the histogram until we are happy we are capturing all true positive T cells, including the weak ones, without capturing any of the background staining that's present. During the algorithm QC process, we will do this for each marker to ensure an accurate level of detection throughout. Once we have set thresholds for each marker of interest, we can begin to QC and confirm the results on the module. Using the co-occurrence matrix, we can look for co-expression of expected markers and look for mutually exclusive markers to ensure we don't have a high frequency of co-expression of these in any of the detected objects. Using the interactive visuals from this, we can easily go back and adjust thresholds where required. We can then visualize all phenotypes generated in the module and view these in combination with each other. As we required visualization of these labels on the image overlays, we took the thresholds defined in the module and incorporated them into the app author to phenotype all objects. We performed summary data analysis using the spatial phenotyping data generated on Visiopharm. The top right plot shows T cell frequencies, and spatial analysis was performed using PhenoExplore, which is an OracleBio proprietary software for spatial analysis, which allowed us to assess differences between macrophage subtypes and T cells. From the results, we can see a much smaller distance between these cells in the glioblastoma tumour and invasive margin ROI compared to the healthy tissue ROI. Finally, using the area quantification data generated on Visiopharm, we can also visualize the data in graph form, which confirms that there was a much greater astrocyte density within the invasive margin ROI in comparison to the healthy or tumour tissue ROI. I'd like to finish by thanking you for your attention, and I'll now pass back for the Q&A where we can take any questions.
Thank you, Karen. Thank you, Gabriel. Hey, guys. Good to see you.
Hi.
So thank you for sharing these insights with us. And thank you, everyone who is watching right now. I think we can start with the Q&A. I am going to start myself with a question. In terms of algorithm development, what would you recommend as a best practice in terms of annotations and resolution, number of iterations, and etc.?
Yeah, I'll take that one. Good question. And I guess there's probably not one size fits all. It's very study dependent. It's very dependent on what type of images, the staining, and what data you want, I guess, out of the images. But I would say in terms of resolution, the rule of thumb we use is that we want to use the lowest magnification possible to detect the objects of interest. And we would typically go, if we were just looking for a tissue find, it would be 2x magnification. And when we want more detail from the classifier, we could go into 10x, and then cell detection will be around 20x, just to get that level of detail. Annotation-wise, we want to have a representative set of images to capture the heterogeneity of if you've got different indications or different staining patterns. You want to make sure your ROIs have the spatial context to capture diversity, and so that your algorithm knows that it's seeing maybe stroma next to tumor, and try and balance the number of classes, balance the number of annotations per class as well. And another point, I guess, the iterations that we train it to, we do check as we go through just to see how it's performing at different points along the training. Again, it's study dependent, but we aim to look for the loss curve to stabilize and converge around 0.1, and use this to gauge the performance of it.
Okay, cool. Thank you. So I'm gonna read question from Max right now. So in relation to the first case study, how do you ensure performance with so many samples? Do you do random spot checks, check outliers, or another method?
Yeah, I'll take that one. So yeah, that's a good question. And that's true, because the full study cohort was over 2,000 images for this study. That said, for the development of all the apps that I showed there, the classifiers, tissue classifiers, and the cell detection, all this development was based on the first batch of images that we received. That was around 600 images. So based on that, we took our standard practice, which is take about 10% for training. So we took around 60 images, we annotated them, or a pathologist annotated them, and we trained the algorithms, as Karen commented there. And then we did the validation on 5% of these images. So again, we run the apps, and then we spot check them to assess performance. We ran some dice scores as well to check the overlap between the manual annotations and the pathologist annotations and the performance of the classifier. In terms of checking all 2,000 images, then we didn't check them all manually. What we did were spot checks, and also based on the types of artifacts that we encountered, just by checking the images when we got them through, like, sent to us, then made the algorithms really robust to detect those. And the two examples I showed during the webinar there, during the first case study, was not just the usual out of focus and tissue faults, but also these bands that we got, like where the scanner lamp failed, basically, and we got these black lines across the images, and also lack of staining. So these were two artifacts that we weren't expected, and we captured during these spot checks when we checked the images, and then we accounted for these artifacts in the apps, in this case in the tissue finds. So then we could flag these artifacts for either manual review or for, to discard the images altogether from the analysis cohort.
Cool. Thank you. Question from Regan. Approximately how long does it take to set positivity thresholds for each biomarker and also for the whole panel? How do you determine true positivity for each biomarker?
Yeah, so again, it's very study dependent. It depends on the images we have, the number of markers. Using the QC tool on and Phenoplex, the QC module, being able to group together your images and see where the staining falls. You've got mean intensity data as well that you can use to sort of identify the mean of the staining marker, but it can take a while. It depends on the number of images. Being able to capture the diversity, I guess, across the cohort. We do a lot of visual QC checks, and assessing it, I guess that way. Yeah, I'm not sure, did I answer the whole question there?
Oh, great, thank you. So question from Bettina, how do you assess which phenotypes or marker combinations are more prominent or relevant in a high-plex scenario like the one presented in the second case study?
Yeah, I can take that one. That's a really good question, because this is becoming more and more relevant because we get more and more clients asking for this, to analyze these high-plex panels. So we have two strategies, basically. The first one would be the client will suggest a specific list of phenotypes. So then we can use tools like Phenoplex to go into, a set of images and then set thresholds manually based on the list of phenotypes they want. And then based on these visual checks on the Phenoplex workflow, we can generate robust data from that. On the other hand, we could have a case where the client has, for example, used an exploratory high-plex panel, and they say, well, that's our 50 markers, so go and find what's relevant here, because they don't necessarily know which combinations will be relevant. So for that, we offer an unsupervised clustering workflow to determine basically which combinations of markers are prominent in the samples that we get. So to do that, we start from the object data that we generate from Visiopharm. So we do our analysis, cell detections, mean intensities per channel. We generate object data from those, and then we put these CSV files or TSV files into our own script, which was developed in our language. And from that, we do some dimensionality reduction, and then we do some unsupervised clustering. And from this, we get a heat map that shows you the expression for each marker in a number of clusters, depending on what the data looks like. So then from these heat maps that we get, we have some strategies to metricize what we get from that to then basically determine in a quantitative way which markers are relevant for each cluster. So then we could suggest, well, the combinations of CD3, CD8, the PDL1 is relevant, and then another cluster would be such and such markers, and so on. So we can do that. We believe Phenoplex will include some unsupervised clustering capabilities in the future, so we are quite excited to test that when it comes out. So,
Cool, exciting. So Sonia asked, how do you ensure that the thresholds set within the algorithms are accurately detecting the specified phenotypes across a full study cohort?
yeah, data one.
Yeah, so phenotyping on a full study cohort is again dependent on the staining variance. We generate object data and use the QC results for exploring any outliers, and this is a instant flag for any additional QC that we might need to make on certain images for certain markers. In Phenoplex, we use the threshold sets, so we can set up the thresholds based off the first image that we're looking at, and apply this on other images to check if the thresholds set for that first image will work across the other ones, or if not, we can amend the threshold set and visually check that it works, or go back and amend the thresholds. Using the mean intensity data also helps to assess it. We can also look to setting different thresholds in different apps, so we can go back into Visiopharm if we want to see the overlays and have low, medium, high threshold set, or an app that detects low intensity cells and an app that detects high intensity cells. But quite a lot of our QC is visual analysis, visual inspection, sorry, of the analysis. And we also, after rendering the data, we will do a QC check of this data. And if we see any outliers from there, we can then go back in and check that the thresholds have been set appropriately for the phenotypes that we need.
Cool, great. And Fabian is asking, how did the guided workflow change your workflow for multiplex analysis?
Data one, or I can talk a bit to that, I guess. So that's a good question, because I guess the way we did that in the past would be we detect the nuclei, and then we need to set thresholds for each marker to define the phenotypes or the populations of interest. And it would be a very back and forth process where you run your app, you need to set your thresholds, you run it on a field of view, then you would check you would say, well, that's a bit too high, so we need to lower the threshold. So then you had to go back to the app, change the thresholds, run it again, go back to the image, and so it was an iterative process, which was fine, but it was not the most efficient. Whereas now with Phenoplex, we can have our image live, we can set the thresholds on the histogram, and everything changes basically live on the image. So you see your dots where that define positivity for the cells. So that's a big plus of the Phenoplex guided workflow efficiency, I'd say, mainly.
Cool, good to hear. Now we have a question from Rasmus. Do you have a strategy for coping with cells that are wrongly phenotyped by the positivity thresholds? And do you know if those cells actually have any significant impact on the study conclusion?
Yeah, I guess it depends on why they've been wrongly phenotyped. Is it an artifact? Is it a staining artifact, or is it maybe bleed through from a different channel, or autofluorescence within the cell body? If it is something like that, if it's autofluorescence or bleed through, we do have steps that we take to counter the detection of those kind of cells. Sorry, it's especially useful if we have like an autofluorescence channel that we can subtract from the rest of the marker channels. If it's maybe there's a biological relevance for it, that we're guided by the clients on that we work with to sort of indicate to us what a positive cell should be, what thresholds they're expecting, or if there's background, being able to distinguish between background and true positivity. We are heavily guided by our clients on that in some instances. But yeah, I guess it depends on the nature of the detection and why it's potentially not a true positive cell.
Great, thank you. Valerie Folk is asking, can we expect the pipeline shown in the first presentation to do the analysis of multiplex images? What are the main limiting factors? Is there work in this direction? Thanks.
Right, so I have to say the pipeline that we set up for the first case study, we need to frame that into potentially into a clinical setting. So that's a retrospective clinical study run by the University of Glasgow and then some commercial partners, as I mentioned, and we are in charge of the digital pathology package. So the idea behind this project, in 3-5 years time, is to have something that could be used in a clinical setting in a hospital with high throughput image workflows, basically. So multiplex is not widely used in clinical settings yet, so we wouldn't expect having to deal with, say, 2000 in multiplex images. It could be done, I guess. More work would have to be put into the QC step, maybe, because here we are just checking chromogenic images. If we had this 6-7 plex, then I guess the QC strategy would change slightly, because then you need to start checking every channel independently. And then the analysis would be a bit more complex as well, and we have to use the likes of probably Phenoplex again to then how do you select the thresholds for each marker consistently across all images. So it could be done, but I believe it's without the scope. It's outwith the scope of this project. Yeah, we wouldn't be doing that in the near future, I'd say, at this large scale anyway.
Cool. All right. Thanks. Fatima said, thanks for the presentation. How do you adjust the thresholds to differentiate positively detected cells on different planes from cells with dual expression?
Yeah, I guess that different planes will mean like the cut of the tissue where you've got maybe overlapping cells that are of different lineages. For example, maybe a macrophage and a immune cell overlapping, and the stain looks like they're within the same body. It's a good question and something that we do see quite a lot, and we will take a hierarchical approach in circumstances like that, where we identify maybe the most important cell class, for example, macrophages, and we'll develop the algorithm to detect that macrophages first, and the remaining nuclei we'll detect as a separate class, and anything within the nuclei class, we can then further threshold for the second marker. So we would, I guess, completely remove the macrophages as one class and not threshold for any of the other markers within that population.
Cool. Bettina is asking, can you provide more detail on how you set up deep learning algorithms for detection of different cell lineages that don't fit into the standard depth detection approach, for example, the microglia cell population?
Yeah, sure. So I touched on it very briefly during the case study, and I think again it goes back to needing to understand what data we want to get out of the analysis and what's most important. I just touched on it there in terms of this hierarchical approach, if macrophages are more important. In terms of the setup within Visiopharm, we identify the hierarchy, we identify what cell lineages are important to detect. So it could be macrophages, DAPI, and maybe a T cell, like a CD3 population. We usually start with the base app from Visiopharm to detect these nuclear objects based off the DAPI, and then amend the training regions that we create to capture the stain from a macrophage or the stain from a CD3, and then add these channels into the AI architect list of channel inputs for the training so that we can create 2 or 3 classes that are focused on the stain that's present for these cells. And yeah, that's it, I guess, in a nutshell, how we approach that.
Thank you. So Regan said, thanks for a fantastic presentation. This was truly a masterclass. Beautiful imagery, real science, and meaningful data. Great job. I agree.
Thank you very much. Yeah. Thank you.
Yes, and then we got a question from Mahmoud that Fabian seems to be replying. Do you expect to find an algorithm to combine the images come out from Visium, spatial genomic, and IMC in case of we have the images of the same slides through both techniques? I don't know if that's a question for you guys or for us, but if you want to answer.
No, that's a question for us at the product management.
Oh.
So
Hi, Fabian.
Yes. Hi, nice talk. Thanks, Gabriel, and thanks, Karen. So that
was
really great. So let me briefly answer that now. So we are working with the tissue aligned module that we are having going in this direction to go for full multimodal tissue aligned capabilities across modalities so that we can integrate all these different approaches in genetics and protein research and RNA to make the most out of your data. I hope this helps, and if you need to find out, then just contact us. Thank you.
Thanks, Fabian. Thank you, guys. Let me check the Q&A chat again. Okay, we got one more question from Brit. So from your workflow description, it sounds like you use Visiopharm and proprietary tools. Was this difficult?
No, it wasn't difficult. It was just normally we use Visiopharm to do the bulk of the image analysis, so cell detection, tissue classification, all that, and then from Visiopharm we just extract object data, and all proprietary tools that we have deal with this tabular data format. So we'll have basically a row per cell, and then for each object we'll have the likes of mean intensity per channel, XY coordinates, area, whatever we want to use that's relevant to the tool, and then the tool will take it from there. The only drawback, I guess, is we cannot get visualization of the results, if that makes sense, because then we are abstracting from what we see on the screen to just data in a tabular format, as I said. So basically what we get would be spatial data or summary data based on the object data that we've extracted from Visiopharm. But yeah, it's in the pipeline to have something where you can feed it back to Visiopharm and then visualize it on a screen, the results of whatever analysis we do out with Visiopharm.
All right, thank you. I think this was our last question, and I'm gonna say thank you so much for this excellent presentation and very engaging Q&A session. Thanks everyone who is here for joining and engaging. That's really really nice. And I also want to invite you all for the next masterclass next month, who is going to be presented by Professor Jared Burks from MD Anderson. So if you are in our email communication, you'll get an invite soon, but also you can check the news on our LinkedIn page. We are always posting recordings there. So the recording from this masterclass is going to be available tomorrow if you want to rewatch and share with your peers. So see you next time. Thank you, Gabrielle and Karen. And
thanks for having us.
Thanks very much.
Thank you. Bye.
Bye-bye.
Bye.